Please continue, sir.
When you have added the plasmid DNA, that means no recombinant DNA to the host DNA, there it gets multiplied. Host DNA with the favorable character gets multiplied and it will be passed on to the next generation. There, to facilitate the host DNA, make many number of copies of the favorable DNA. One particular enzyme is responsible for that. It is called DNA polymerase. Polymerase is nothing but many more times. It is replicating many more times. For that, a particular enzyme called DNA polymerase helps that operation in the host DNA. Then, what are the process? Again, now we will once again understand what are the tools in this process. You will cut it, DNA isolation, and then sequencing, identifying the gene, cutting the gene with CRISPR-Cas9, and then a plasmid, you are attaching this cut portion of the DNA, which is gene of interest, to the plasmid DNA, which is a carrier. Plasmid DNA together with the gene of interest together is called mRNA, MDA. This is messenger RNA. Messenger RNA then gets introduced into the host DNA. DNA polymerase helps host DNA to produce many more replications of the host DNA with favorable character. Ultimately, it gets expressed in the offspring. General tools are recombinant DNA happens, then restriction enzymes, plasmid DNA, then host enzyme, polymerase enzyme. These terms you remember. Here, from the same species, same family, if a good trait is identified and then introduced into the animal of same family, species, this is called cisgenic. Same, suppose a good trait in lion family, somewhere you will take out a positive feature from another line, introducing here. That is called a cisgenic mechanism, cisgenic process. You are taking out altogether from a cat, you are taking out a positive, and then now you are introducing into some other animal. This is called transgenic. Trans, cis is same, trans is different. Another thing is called subgeneric. The third kind of genetic mechanism is called subgeneric. Subgeneric is nothing but you are not going to the mother and father, you are taking out one particular gene sequence from an animal or a human being. You are editing it, you are taking out a deleterious part, and then no, you are not adding anything. That means as diseased part is there, a particular gene is unable to express, it is a diseased gene. You are simply cutting it out, taking it out. That's all. It is called subgeneric. That means it is within that. Cisgenic is between the same species. Transgenic is species will be different. Subgeneric is within the same either plant or animal. You are doing out editing something is subgeneric. Well, up to this part, if you have any doubt, you will clarify. You will ask me, I will clarify, because this is the important point to understand the further applications of genetic engineering. This entire process which I told you is called as gene therapy or genetic engineering. For genetic engineering, there are two conditions are required. One, biological, the genetic engineering is one part, chemical engineering another part. Genetic engineering, I told you all the processes, but chemical engineering is added to genetic engineering because you want a sterile atmosphere for doing all these manipulations. Your entire lab, wherever you are performing these operations, should have complete sterile ambience. No, it should be completely sterile, disease free, and then there should not be any other thing. Then only the processes will function, will accurately, you will, scientists can process these functions accurately. Otherwise, interference will be there. Now, up to this, you are free to ask questions.
Any questions? Any questions, dear students?
No, ma'am.
Yeah.
In that case, I will stop here, and then in tomorrow's lecture, we will talk about genetically modified crops.
Yes, sir. Yes.
Based on this technology, I am going to tell you so that now you will be easily understanding. It won't take much time. Whatever time that is to be taken now, let us take now itself to understand the process. But don't worry, tomorrow also, if you have any doubt, no, in between also, I can repeat this information which I have given today.
Yes, sir. So today we
Assuming that you have no doubts, shall I conclude?
Yes, sir.